Hypophysis, a pituitary gland. Hypophysis is a compound endocrine gland suspended from the hypothalamus and located in the sella turcica, a depressions in the bony floor of cranial cavity. A short stalk, the infundibulum, attached pituitary to the hypothalamus. This small gland, which is about the size of a pea, is often referred to as the master gland because it produces several hormones that influence the activity of other endocrine glands. The hypophysis may be divided into glandular portions, the adenohypophysis, and in neural portions, the neurohypophysis. The embryological development of the hypophysis explains the presence of these two distinct tissue types. The adenohypophysis is derived from an outer pocketing called Redke's pouch of the ectoderm of a primitive oral cavity. The neurohypophysis is developed from outpocketing of the neuroectoderm of a primitive brain, especially the diencephalon. The adenohypophysis is further divided into a pars distalis, which comprises the bulk of the adenohypophysis, a pars tubularis, and a pars intermedia. The neurohypophysis is divided into pars nervosa and the infundibulum. This infundibulum forms the stalk of the hypophysis. Additionally, the pars distalis and pars tubularis are considered to be the anterior lobe of a gland, and the pars nervosa and pars intermedia are called as the posterior lobes. The pars distalis constitutes about 75% of the hypophysis and is enclosed almost completely in the dense fibrous capsule. The parenchyma is in the form of anastomosing cords and clusters of epithelial cells supported by network of reticular fibers continuous at the periphery with components fiber of capsules. Between the parenchyma cells are sinusoidal capillaries. The parenchyma is composed of two main categories of cells, chromophores and chromophils. Chromophils are subdivided into acidophils and basophils on the basis of staining reactions of their cytoplasmic granules. However, the dyes used to distinguish these cells are acid dyes and do not distinguish acidic and basic properties of the cells. Many workers have developed the non-committal terms alpha and beta cells for the two types of chromophils. The chromophobe cells, which have little affinity for dyes, sometimes are referred as chips or C cells. The relative proportions of the cells vary markedly in man. Chromophobes constitute approximately 50% of cells, acidophils 35% and basophils 15%. The proportions may be altered considerably by castrations, thyroidectomy, or other experimental procedures. Additional cell types may be demonstrated within the alpha and beta groups by spatial staining techniques and histochemical methods. The cells also may be identified by immunocytochemical staining, using antibodies prepared against any one of the purified hypophyseal hormones. In this way, it is possible to classify the cell types within the pars distalis on the basis of one or more hormones they contain. In the chromophils, it is believed that the granules are actual precursors of secretions. The degree of granularity is correlated with the physiological state of the cell when fixed. The cells are thought to secrete cyclically rather than continuously. Mitosis are seen rarely in any of the cell types, and it is certain that few, if any, of the cells are destroyed when the secreted product is released. It appears that some chromophobes represent reserves or inactive cells, which gives rise to the chromophils. As the chromophobes become active, granules forms within their cytoplasm. The granules are specific for the different cell types. Engorged cells then secrete, and the cells revert to an inactive state. Chromophobe cells, or you can say it's a C cells, these faintly staining cells in the past were known as reserve cells. They are small rounded or polygonal cells with relatively little cytoplasm. The boundaries of the cells are not easily visible in ordinary preparations, and generally, on light microscopy, the cytoplasm lacks specific granules. On electron microscopy, however, many cells exhibit small secretory granules. It appears that most chromophobes are partially degranulated chromophils, and that only a small percentage of them should be considered reserve or neurosecretory cells. The chromophobes often appear in groups in the center of the cord. Now, chromophil cells. In this, we'll see first acidophils, that is alpha cells. The acidophils stains readily and are identified easily in ordinary preparations. They are larger than chromophobes and their cell boundaries are distinct. The cytoplasm is crowded with small specific granules which are stained by numerous dyes such as eosin, acid fused chains, orange G, and azocarmine. The affinity of the granules for the latter two dyes is used to differentiate two types of acidophils. Those whose granules takes up the orange G of an azon stains are called organophils, are alpha acidophils. Those in which the granule stains intensely with azocarmine are carminophils or epsilon acidophils. The organophils are the cells responsible for the secretion of growth hormone known as somatotropins, abbreviated as STH. Thus they are termed somatotrophs by some authors. On electron microscopy, these cells shows an extensive development of granular endoplasmic reticulum and contains a population of electron dense granules which range from 3000 angstrom, that is around 300 nanometer, to 3500 angstrom, 350 nanometer in diameters. Somatotropin stimulates general body growth, particularly growth at the epiphysis of bones. Hypophyseectomy causes a cessation of growth which can be restored to normal by administrations of this hormone. Undersecretions lead to dwarfisms in certain animals, and oversecretions, as in certain tumors of anterior lobe, causes gigantisms in children. If oversecretion occur after closure of epiphysis discs, a condition known as acromegaly, in which the bones become thicker and hands and feet broader. Here these granules are much larger, over 6000 angstrom unit in maximum diameter in carminophils than in organophils, and are more scattered within the cytoplasm. Granular endoplasmic reticulum fills a large proportions of the cytoplasm. These cells, also termed mammotrophs, are concentrated in the postlateral regions of the pars distalis and are greatly increased in number during and after pregnancy. They secrete lactogenic hormones, prolactin, luteotropic hormones, or LTH, which initiate and maintain the secretion of milk after pregnancy and stimulate the corpus luteum of the ovary to secrete progesterone. Next cell is a basophils or beta cells. The basophils tends to be appreciably larger than the acidophils. The granules are less numerous than in acidophils and are smaller, about 150 to 200 nanometer in diameters. They stain rather poorly with hematoxylin but are stained deeply with methylene blue. Basophils are best identified by the periodic acid skip reagent It is called as a pass reactions technique where they are strongly positive owing to the concentration of glycoproteins in their secretory granules. In most mammals, at least two types of basophils can be distinguished by simple staining. One type stains with aldehyde fish chains, that is beta basophils, whereas the other stain with delta basophils that does not stain with these fish chains. They are called as the delta basophils. Here we'll see first beta basophils or thyrotrophs that secrete thyrotropic hormones called as thyroid stimulating hormone TSH, and the cells are relatively large and contain numerous granules, principally concentrated within the peripheral cytoplasm. The granules are dense and small and are large in size from 1000 angstrom to 1500 angstrom or more. TSH maintains and stimulates the thyroid epithelium. Hypophysectomy results in atrophy of the thyroid, which may be restored to activity by administration of hormone extract. Injections of TSH in normal animals produce all the symptoms of hyperthyroidism. Thyroidectomy results in the increase in the percentage of basophils within the pars distalis. Next one is the delta basophils. Delta basophil cells that do not stain with aldehyde questions include the gonadotrophs and the corticotrophs. Here the gonatotrophs are distinguished further into two types on the basis of their granular content. They are gonadotrophs that possess abundant granules, about 2000 angstrom in diameter, a well-developed Golgi apparatus, and irregularly distinct cisternae of granular endoplasmic reticulum produce the follicular stimulating hormone, that is FSH. Gonadotrophs of a second type secrete luteinizing hormone and are small, spherical cells that contain uniform, dense granules about 2500 angstrom in diameter, usually concentrated in one side of the cell. The Golgi apparatus is not as extensive as in FSH producing cells, and the granular endoplasmic reticulum is composed of flattened element. Next one is a corticotrophs. The corticotrophs are larger basophils that contain only a few scattered membrane-bound granules about 2000 angstrom size in diameter, an eccentric indented nucleus, and an extensive Golgi apparatus that occupies large portions of the cell. These cells are responsible for the secretion of ACTH, adrenocorticotropic hormones, a peptide which promotes growth of a suprarenal cortex and stimulates secretion of glucocorticoids. Next part is the pars intermedia. In man, the pars intermedia is less well developed than in many other animals and usually is poorly defined. It forms only about 2% of the hypophysis. It is composed of a thin layer of cells and of vesicles that contains colloid. It lies in close relation to the residual lumen, which virtually is obliterate in most adults. Some component cells, polygonal in shape and small and pale staining. Others are somewhat larger and granular and stain deeply with basic dyes. The cells which are basophilic possess an eccentric nucleus resembling corticotrophs of the pars distalis and frequently extend as cords for short distance into the pars nervosa. The cell lining the colloid contains vesicles, commonly are ciliated, and some are mucus secreting. In man, it is not clear at present whether MSH is synthesized by basophils of the pars intermedia or by corticotrophs within the pars distalis. Next is the pars tubularis. The pars tubularis forms a collar of cell around the infundibular stalk. The cells, in close association with numerous blood cells, are arranged in groups or short cords longitudinally oriented. They are cuboidal, and the cytoplasm, which is weakly basophil, contain fine granules and some glycogen. Small vesicles which contain colloids are seen occasionally. The functions of pars tubularis, if any, is unknown. Next one is a neurohypophysis. The cell of neurohypophysis produce two peptide hormones, oxytocin and antidiuretic hormone ADH or vasopressin. The release of this substance into the systemic circulation is regulated by the hypothalamus and higher brain levels. General functions of hormones are like stimulations of contractile cells in mammary glands, the decreased urine volume causing the kidney to remove water from newly formed urine. It should be noted that even though oxytocin does stimulate smooth muscle contraction in a pregnant uterus, this may not be a normal physiological functions of a hormone. ADH basically affects the permeability of distal convoluted tubules and collecting ducts of a kidney and thus controls water absorption from these ducts. Individuals producing low level of ADH excrete large amount of dilute urine and suffers from extreme thirst, that is called as a diabetes insipidus. The neurohypophysis consists of the post lobe and infundibular stem and the median eminence. It contains pituitary an axonal process of unmyelinated nerve fibers, whose cells bodies are located in the paraventricular and supraoptic nuclei of the hypothalamus. The axonal process, approximately 1 lakhs, converge at the median eminence, forming a bundle of fibers known as hypothalamo-hypophyseal tract and pass through the infundibular stem to reach the pars nervosa. These nerve fibers are unusual in that they do not end on other neurons or effector cells, but end blindly close to vessels of a rich capillary plexus of pars nervosa. As was noted earlier, the capillaries of hypophysis are fenestrated. The circular fenestrations in the attenuated endothelium are closed by thin diaphragms. The terminal portions of axonal process are extended and commonly contain numerous secretory granules, around 100 to 300 nanometer in diameter. They are called as herring bodies. The neurosecretory substance contained in these granules are believed to be synthesized in the cell bodies and then transported through the axons to the nerve terminals where they are released in response to hypothalamic nerve impulses. The released hormonal substance transfers the perivascular space, enters the fenestrated capillaries, and are distributed to target organs, tissues, and cells via the systemic circulation. It should be noted that the neurohypophyseal hormones like ADH and oxytocin are found to carry proteins called neurophysin during the axonal transport and storage in a pars nervosa. This hormone neurophysin complex constitute the bulk of the herring bodies. The hormones are freed from the neurophysins when they are released from the nerve terminals. The pituitary resembles the neuroglial cells found in the central nervous system, except that the pituitary may contain pigment granules. The nuclei of the pituitary are round or oval, and the cytoplasm is drawn into processes that are often associated with the axonal processes and terminal expansions of the nerve fibers. But it is unknown if the pituocytes have metabolic activities associated with the hormone secretions.